This is a short discussion just going over some of the techniques that are used to gather data for evolutionary developmental biology. By no means is this all of them, but we will talk about some very important ones. The first one is gene visualization. Now, when we say gene visualization, realize what we really mean is gene expression. So this is a picture, a couple pictures actually, of fruit fly embryos. And just as in your body, every cell in a fruit fly's body gets the entire instruction manual. So every cell has all of the genes. But different cells are using different subsets of those genes. So some cells, like in the muscle, might be using lots of myoglobin and lots of respiratory cell genes and lots of other things, whereas a brain cell might have a very different profile of genes that are being used. And in development, this is especially important because, as we talked about with those transcription factors, the particular genes that are being expressed and turned on have a dramatic impact on what those cells are going to become. So if we can figure out what genes are being turned on, and maybe even the order in which those genes are turned on, essentially we have a recipe or a set of instructions for how to build a specific part of the body. And what we would see is that we have a different profile coming on in the head than we do in the middle part of the body. Or at a later stage, we might see something like nerve cells expressing this gene where the rest of the cells are not. So gene expression visualization is critical. Now, there are a couple of methods by which this is accomplished, and we're not going to go into any detail, but I'm going to list two of them. In fact, they're important enough that I'll write them here. So we have an antibody stain, which uses proteins that recognize the target protein that you're looking at. So antibodies bind to many things, but one of the things they can bind to are proteins. And so if we want to look at a specific gene product, a protein, then we can create an antibody that recognizes that and then look to see where that antibody is. The other thing we can do is we can do an in situ hybridization. Now, we've actually come across this before because when we use fluorescence, it's called FISH, which is an acronym for fluorescent in situ hybridization. So this diagram on the right is a FISH where we have three different fluorescently labeled products. And notice that these ones in red are very, very different from the ones in blue, and they're expressing different genes. And so a FISH, rather than targeting the protein product of a gene, targets the mRNA transcript. But they're both looking at gene expression, just slightly different steps in the gene expression process. Okay, so just be aware of both of those, that both an antibody stain and an in situ hybridization are ways that we can look at gene expression. Now, those are only giving us ideas about what's occurring, but without any manipulation or any change in the genes themselves. So, especially in model organisms, human researchers have used another technique to look at what happens when we change gene expression. So knockout is a way to turn off or maybe just greatly tamp down or turn down levels of expression. And so it's a way to make artificial homeotic mutations. If we turn off UBX, we end up growing wings on both the middle thoracic segment, where flies usually grow their pair of wings, but also on the hind thoracic segment. So that is a knockout expression, and it shows us the impact of not having a gene there, or at least not having it at high levels. Ectopic expression is a way to express a gene in the wrong location, or at least in a location where it's normally not expressed. And so you may be able to remember this one easily by remembering ectopic pregnancy. An ectopic pregnancy is a pregnancy where implantation of the embryo occurs in the wrong place, usually in the fallopian tubes. And that can be quite serious and have a dramatic outcome. But ectopic simply means at the wrong place or in the wrong location. And so when we ectopically express a gene, like a gene that controls eye development in the wrong place, we may grow an eye. And this is interesting because if we express the right gene, a very early gene in the whole cascade of how to build an eye, we can actually turn on everything downstream. So by just ectopically expressing that one gene, the next set of genes are turned on, then the next set, and we end up growing nearly a fully formed eye in the wrong location just by turning on that one gene. And so we'd have a good idea about the role of that gene. Is it a gene that kind of directs the entire expression of an eye? If if we get a nearly fully formed eye, then the answer is yes. Otherwise, maybe if we only get lens development or if we only get nerve growth or something, then we know that it plays a role a little bit more downstream and is maybe not so critical in the formation of an eye. And this actually gives us another insight into this concept of homeotic genes, the ones that make the most dramatic change, where we can grow an entire leg or we can grow an entire wing where normally we'd only get halteres, are ones that are very, very early in the process of how to build a wing. So think of them kind of as the master control switches, and we turn one on and then other genes are turned on. So that one little tiny change, and it gives you an idea of how you can have a dramatic impact downstream with just a small genetic change. And then finally, one other very critical thing that also adds one more bit of data to helping us understand our hypothesis is this idea of a transgenic expression. So technically, transgenic is a gene that's from a different organism expressed into a fly. So for instance, we could take a mouse gene and express it in a fly and look to see what happens. And one of the things that we found is with some of these homeotic genes that they're actually swappable. So for instance, this gene here that was ectopically expressed to grow a fruit fly gene, if we knock out that gene, you don't get any eyes growing at all. So you would get not only no eyes and antenna or anywhere else, but the eyes where they were supposed to grow would not grow because we had basically turned off or even removed, in some methods like in fruit flies you can do that. We've removed that gene so those organisms no longer have it. Now in most organisms, loss of an eye is a critically, even a fatal mutation, but you can take care of them in the lab and you can breed them and you can actually create lineages of flies without any eye development at all, just by turning off this one gene. Now, here's the key, the link in with transgenic expression. If we then take the mouse gene that builds mice eyes, there's the same gene found in mice that helps to build their eyes, and we replace the mutant or the lost gene in fruit flies with a mouse gene, you can actually recover eyes, and they're not mouse eyes. It's just the fruit fly can use that same mouse gene, even though it's separated by 300 or more million years of evolution. Actually, going back probably more like 500 million years of evolution. Even though there's been those many millions and millions of years of evolution, these genes are still conserved enough. The same gene in fruit flies and the same gene in mice are basically swappable. You can take the mouse gene, put it in the fruit fly in replace of a defective one, and everything works just as it should in the normal fruit fly. So this really got people thinking about these genes and thinking them almost as like tools, like a hammer, right? If I took a hammer from a carpenter's kit and gave it to a mechanic, the mechanic could use it to do whatever he does with a hammer, right? Maybe knock out a dent in a fender, or to help loosen up a piece of an engine that needed to be loosened up. Whereas a carpenter might use it very differently, like to drive a nail. So these genes can be used, and oftentimes they have similar functions, but we really start to think about these genes as tools. And what we found is that these tools are incredibly conserved. You find the same genes in you that you find in a fruit fly. So remember the Hox genes? All animals have them. And you have many of these same genes that a fruit fly has. And so this leads to some really important conclusions that we'll talk about and wrap up in our last discussion. That's 3.3D. But before we do that, I just want to give you one example of how understanding these changes in these genes can lead to an understanding of the evolution of a group. And we're actually going to use plants and flowering plants for this. One of the best understood structures in plants is the flower. It's really critical and was a key innovation that allowed them to do really, really well. And there's lots of diversity and variety in flowers. And so people began to study some of the earliest, simplest flowering plants and then look at the more complex ones. And what they found is that in the very earliest ones, they only had a couple of genes, and these genes basically could only define two different flower parts. And so the earliest flowering plants were very simple with just two basic parts. But then later they gained additional copies. AP3 is a duplicated version of AP1. Sep is a duplicated version of pi is a duplicated version of sep. And so we start to get these very complex forms with different combinations of genes. And then when we map those onto our phylogenies, we can actually locate where those changes occurred. So the very earliest flowers have duplicated genes. We'll talk about gene duplication more in the next unit, but those genes are still very, very similar. So basically you have extra copies, but they don't do anything different. So you get this very simple flower with just 2 parts. Later, one of those duplicated versions gets a brand new motif, meaning it has new characteristics, new mutations that the ancestor didn't have. And so now we have 2 distinct versions of this very distinct gene and another one that gains extra gaps, and now we can build slightly more complex flowers. Okay, but still not really complex, but now we've got 3 basic parts instead of 2 basic parts. And then later we end up with another duplication yielding a 3rd gene with then subsequent diversification and mutations occurring in that give us even more complex flowers with 4 or more parts. Okay, and so by understanding those genetic changes, using the tools that we've talked about by where they're expressed and turning them on and off and seeing what the impacts are, we can identify their role, and then mapping where those changes occurred by looking at what developmental genes these ones have versus these ones, mapping them and then mapping the changes. We can really get a very nice complete picture of these critical developmental changes. And this is just one example in plants. We've done the same thing for insects and how they derive from crustaceans, or mammals and how we have a development of limbs in mammals, and then how limbs change so the forelimbs are different than the hind limbs. And these have been mapped down to sometimes single mutations or small groups of mutations in these critically important developmental genes that lead to those differences. But remember, many of the genes are still the same, and we showed this when we used these methods to show you that a fruit fly gene that can also be found in a mouse often have very, very conserved role. And so we're going to begin to revise our hypothesis, and we'll talk about that and give you the final revision after we talk about another few key concepts in section 3.3D.